everyone, I'm Nicole and you're watching BioCorner. Today we're going to be talking about a gene editing tool I've actually used in my own research and has been in the news quite a bit the last few years. That's right, we're going to talk about CRISPR. Let's brush up on the basics of our cells before we dive into the complications of gene editing. Our cells are constantly making proteins, which are like little biological machines that do all sorts of work around the cell, like making energy from sugar and helping cells grow. We can imagine the cells in our bodies are kind of like biological Lego cities. There are tons of buildings and cars that make the city run, and those are the proteins. And each building or car needs instructions on how to put the bricks together to make them. And those are the genes. Put all of the instructions or genes together in one place, and that is the DNA of the cell. Okay, so now that we have all that covered, let's dive into gene editing. CRISPR is short for CRISPR-Cas9. Cas9 is a special protein found in bacteria that can cut DNA and is part of the bacteria's immune system, so it helps them fight off diseases. CRISPR is an acronym for a really long name and is basically a short bit of genetic instructions to help direct Cas9 to the specific part of a gene it should go to and cut. You can think of CRISPR as the control-find feature on a computer. Researchers like me can then program CRISPR to target a particular gene in DNA to be cut and then modify it. So we know what genes are, and we know CRISPR is a tool to edit them. How exactly does CRISPR work? I'm going to show you using some blocks. Okay, so here we have the specific DNA we'd like to edit. It's normal and unedited. Now, along comes CRISPR, which in this case will just be my hands, and it acts like a pair of scissors and cuts our DNA, making what is known as a double-strand break. Your body has two options for how to fix this. One way is for it to try and put everything back together exactly the way that it was. The only problem is that because of some slightly complicated chemistry that we won't get into here, the DNA can't just be glued back together like this. Instead, it has to take one or two blocks off first. Once it does this, it has to try and remember exactly what blocks were used and how many. And, to your body's credit, a lot of the time it gets this right. It's able to put back on the one blue block and the one yellow block and glue it back together exactly the way that it was. But sometimes it messes up and forgets a block or adds an extra one. And either way, this majorly messes up the instructions. It would be like if someone ripped out the second page of your Lego or baking instructions, but still expected you to make the right thing? You can't, right? You can try and fill in the missing page from memory or by guessing, but you won't get it right every time. Well, the same goes for your cells. They often end up making a faulty, useless protein when they try to repair this cut that CRISPR has made. And scientists can use this method to disable certain proteins and study how the cell behaves when they aren't around. Now, remember that I mentioned there's a second way to fix the cut CRISPR made? So the second way, which I'll call the copy and paste method, starts off just like the first. You have your normal, unedited DNA, CRISPR comes along and makes a cut, and your cell has to take off a couple of blocks. But this time, instead of trying to remember what the original instructions were, we give the cell a little bit of help and give it brand new instructions to copy and paste into the DNA. So instead of adding back a blue and a yellow block, we add on two red blocks. A lot of times, scientists use this technique to introduce a specific change, also known as a mutation, to the DNA in order to understand what that mutation does. It would be like swapping salt for sugar while baking, or for anyone that is a fan of the TV show Friends, like when Rachel made a traditional English trifle.
It's a trifle. Raspberries, more lady fingers, then beef sauteed with peas and onions.
The pages are stuck together.
In this case, the proteins are still made, albeit differently, and still tries to do its job. Depending on the mutation or change to the DNA sequence that was made, the protein will either do the same, better, or worse at its job. It's like when you make a mistake while cooking. You can argue it's still edible, technically,
It tastes like feet.
I like it.
So scientists can use this copy and paste method either to introduce a mutation in order to understand it more, or try and fix a mistake that is already there and change the instructions back to what they're supposed to be. And this has all kinds of applications, from agriculture to medicine, and I've posted a few interesting articles about these applications in the description if you want to check those out. Now, we still have a lot to learn about this tool and how it can be used safely. But for now, it is helping scientists better understand some really complicated things in our bodies, and that alone is really great. Okay, that's all I have for now, guys. Make sure to like and subscribe if we've earned it, and post in the comments something new that you learned, or if there's a topic you want covered in another video. Bye!